welcome to the deep dive. Today we're really getting into something critical for pharmaceutical research, targeting G protein-coupled receptors, GPCRs, right?
Exactly.
We've been looking into Tanso Biosciences. They seem to specialize in providing researchers, perhaps like you listening, with, well, better tools for this area.
That's right. I mean, so many important drug targets are GPCRs, but working with them isn't always straightforward.
Hmm.
So yeah, our goal today is to figure out how Tanso Biosciences services might actually help you speed up your drug discovery. They've got this focused set of technologies based around GPCR functional assays.
Okay, let's unpack that a bit. So it looks like they're doing their own drug discovery,
What?
especially for these orphan GPCRs, the ones where we don't know the activator.
Yeah, that's a big part of it. But they also offer contract services directly to pharma companies.
Got it. And the core seems to be this unique sort of in-house tech for analyzing GPCR function comprehensively.
Right. And what's really interesting, I think, is the detail their technology can apparently capture. They use these proprietary reporter cells, which lets them measure the signaling from the GPCRs with high sensitivity and specificity. So you get a much clearer picture of what the receptor's actually doing compared to maybe some older methods.
That sounds useful. And crucially for researchers using different models, you mentioned their assays aren't just for human GPCRs.
No, not at all. They can handle GPCRs from humans, mice, rats, rabbits, dogs, even macaques.
Wow, okay.
Plus, it seems like they get that research isn't one size fits all. They offer customization, for pharma, biotech, but also food science labs, academic groups.
Right. Tailoring it.
Exactly.
Yeah.
Their whole philosophy seems to be about giving drug discovery better tools, specifically for GPCRs.
Right.
And given how many GPCRs there are and how vital they are, well, that focus could be a real advantage.
Okay, so let's get down to brass tacks. If I'm a researcher listening, trying to push my project forward, what are the immediate wins using Tanso's platform? The scope, the sheer number of GPCRs they cover, that sounds like a big one.
It really is. We're talking functional assays for over 550 human and mouse GPCRs. Yep, roughly 300 human, 250 mouse. And importantly, if you're looking at orthologs or maybe a less common target, they say their system is built to expand quickly.
So comprehensive target coverage, as they put it, means you're more likely to find what you need fast.
Pretty much, yeah.
Now, this is where, for me, it gets really interesting. They claim their platform can measure signaling through all four major G protein families, Gs, GO, GQ11, and G1213.
Mm-hmm.
Now, I understand G1213 has been, well, a bit of a headache technically. Why is being able to assay that pathway such a big deal for drug developers?
That's a fantastic point because historically, yeah, reliably measuring G1213 signaling was tough. It means a whole group of potential drug targets, GPCRs, coupled to G1213 might have been, sidelined or harder to study.
Right.
So Tanso saying all G protein subtypes, no exceptions. If they've genuinely cracked that, it could open up totally new ways to tackle diseases where G1213 is involved. We could be looking at new treatments for things where options are currently limited.
That really could be a game changer for researchers in those specific areas. Okay, what about the basics? Reliable data. How good are the assays themselves? Sensitivity, specificity.
They emphasize ultra-sensitive and specific detection. What that means for you, the researcher, is you can reliably pick up even quite small biological effects from your compounds acting on the GPCRs.
Okay.
And you can do that with minimal background noise confusing the results. So if you're screening thousands of compounds or trying to understand subtle differences between lead candidates, that kind of reliability is, well, it's essential for making good decisions.
Makes sense. And in pharma R&D, speed is always critical, isn't it? How flexible are they?
Their approach is described as fast, flexible, and client-centric. They seem keen to adapt the assay design to fit your specific research question.
So not just off the shelf.
Exactly. Need rapid, high-throughput screening, they can do that. Need detailed SAR studies, that too. Need to check how your compound works across different species. They say they can tailor the service to hit your timelines and goals.
Now, something that really stood out to me was their work on orphan GPCRs. Could you explain for listeners why that's such a hot but challenging area and what Tanso's doing there?
Absolutely. Orphan GPCRs are this huge, untapped reservoir of potential drug targets. We call them orphan because we haven't found their natural endogenous ligand yet, the key that normally unlocks them in the body.
Right, so it's hard to know what they do.
Precisely. Which makes it tough to figure out how to target them therapeutically. Tanso is actively trying to de-orphanize these receptors, find those missing keys. They have their own internal drug discovery programs looking at about 100 orphans,
Wow.
screening libraries to find agonists, activators, prioritizing ones that might be linked to diseases.
So by finding these activators, they're basically putting these unknown receptors on the map for drug development.
That's the idea. And they've already had some success. They've identified agonists for 6 specific orphan GPCRs: GPR39, GPR55, GPR65, GPR68, GPR132, and P2R White 10. And the work doesn't stop there. They're characterizing these agonists further and using them to develop assays to find antagonist blockers for these newly validated targets. So this orphan GPCR expertise feeds right into their wider drug discovery efforts, could lead to some first-in-class drugs down the line.
It's crucial, though, that the tools researchers use are scientifically sound. Is Tanso's technology validated? Protected?
It seems so. They mention international patents, specifically WO 2000-06979, which suggests the tech is novel.
Okay, that's important.
Yeah, the scientifically validated, globally protected status gives some assurance. Plus, they mention it's being used in academic and industry research collaborations, which is another form of validation, really.
Right. Okay, let's get super practical for the pharma researcher listening. What are the specific day-to-day ways they could actually use Tanso's assay platform?
Oh, there are quite a few applications, really covering different stages. You can use it for your main on-target screening, obviously confirming your compound hits the receptor you want it to hit. But also, crucially, for off-target screening. Checking if your compound accidentally hits other GPCRs. That's vital for spotting potential safety issues early.
Yeah, catching those off-target effects early must save a ton of time and money later. What else?
They can help with direct cross-species comparisons. So you can see how your compound acts on, say, the mouse version versus the human version of your target, essential for translational work.
Right, predicting human effect.
Exactly. They do detailed compound profiling SAR analysis to help you optimize your leads. The data could even help if you're thinking about licensing in a compound or maybe repurposing an existing drug. And they offer both big panels and smaller, focused ones.
And in terms of the type of activity you can measure?
Pretty comprehensive. They can generate concentration response curves to show agonist activity, antagonist activity, inverse agonist, even measuring modulators, PAMs, and NAMs.
Okay.
And they also have this quite unique capability to look at pH-dependent activity, how the receptor functions at different acidity levels, which can matter in certain tissues or disease states. So the uses are really quite broad.
Let's dig into the different assay formats. What choices does a researcher have?
They offer several assay types. There are the big panel assays screening against hundreds of human or mouse GPCRs at once.
For broad profiling.
Right. Or, for a more focused approach, there's the Flex Panel 48. You pick any 48 receptors you want.
Any 48?
Yep. Either a custom mix or they have preset panels focused on 13 different therapeutic areas: immunology, oncology, neurology, cardiology, you name it.
That sounds efficient.
And critically, they also have specific safety panels. A standard one with 24 GPCRs, often linked to side effects, and a premium one expanding that to 55 GPCRs for broader safety checking.
Those safety panels seem like a really smart tool for de-risking. What other formats?
Well, beyond panels, they offer detailed concentration dependence analysis where you can customize the setup, like number of replicates and concentrations. For animal model work, the ortholog assays let you test GPCRs from pretty much any species you need.
You mentioned species differences earlier.
Yeah, they show examples where ligands have quite different potencies on, say, human versus rat FPR1 or human versus mouse C5AR1. Important stuff to know.
Definitely.
And finally, those proton sensitivity assays we mentioned for looking at pH effects on receptors like GPR132 or GPR68.
Seeing some concrete examples always helps bring this stuff to life. Any highlights they shared?
They did. They showed how carvedilol, screened against 200 human GPCRs, selectively antagonized just a few adrenergic receptors, as expected.
Okay, showing selectivity.
Exactly. They showed valsartan's concentration-dependent block on AGTR1. They highlighted those species differences again. For the orphan work, they showed a dose-response curve for a new agonist activating GPR68, and GPR65's activity changing with pH. They even showed data for LPR6, one of those tricky G12-313 coupled receptors.
And those Flex Panel 48 examples.
Right. They showed the diabetes drug candidate TK875 selectively hitting FFAR1 in their endocrinology panel, then followed up with the full dose response.
Hmm.
And similarly, naloxone selectively blocking opioid receptors in the psychiatry panel, again followed by concentration-dependent analysis. These assay examples really demonstrate what the platform can do.
You touched on the GPCR safety panels again there. Let's just clarify. For researchers really focused on safety, why are these panels so valuable?
They're specifically built to help you spot potential trouble early. We're talking about the drug discovery and preclinical stages.
Before things get too expensive.
Precisely. By testing your compound against GPCRs known to be frequently involved in adverse drug reactions, you get an early warning signal. The standard panel gives you a solid baseline with 24 key receptors,
Okay.
and the premium panel gives broader coverage with 55. Finding these unintended interactions early lets you make smarter choices about which compounds to move forward, or maybe guide chemists to design out the problem.
This definitely sounds like a comprehensive toolkit for, smoothing out and reducing risk in GPCR drug discovery. So, if a researcher listening is thinking, okay, this sounds interesting, how do I learn more or even place an order? What's the process?
The best starting point is probably their website. All the details about the services and assays are there. They have an inquiry and RFQ form request for quotation right on the site.
So you can outline your specific need.
Exactly. Fill that out to start the conversation and get a quote. Or for a more direct approach, you can just email them, info@tansobio.com. Their team should be able to answer questions and guide you.
And are there resources online they can browse first?
Yeah, absolutely. The website has a full GPCR catalog. It lists all the human and mouse receptors they currently offer, often with example concentration response curves. That could be really helpful for planning.
Good resource.
And as we mentioned, if you need a GPCR from a different species, they say they can custom prepare those on demand. So lots of flexibility there for cross-species work.
Okay, so summing up, Tanso Biosciences seems really dedicated to helping pharmaceutical researchers like you, the listener, with these super sensitive, broad, and adaptable tools for GPCR drug discovery. The aim seems to be faster development, lower R&D risk.
I think that's a fair summary. The comprehensive coverage, hitting all G protein subtypes, the high sensitivity, the flexibility, plus that special expertise in orphan GPCRs. It looks like a potentially valuable partnership for tackling GPCR targets.
So if you are a researcher working on GPCRs, it definitely sounds worth checking out the Tanso Biosciences website or just dropping them an email again. That's info@tansobio.com. See their catalog, explore the services, and maybe see how their platform could fit into your next project.
Which leads us to maybe a final thought for you to consider. Thinking about your own research right now, where are the bottlenecks? What specific challenges with GPCR assays could a platform like Tanso's potentially solve? And perhaps more excitingly, what new avenues, maybe even new targets, could you explore if you had access to this kind of comprehensive and specialized technology?